Hello guys, welcome back to our biochemistry lecture series. So today's our discussion is about nucleic acids, and to understand this topic I suggest you to please first watch my previous videos because to understand this video you need some little basics which I discussed in the previous videos. So I hope if you will first watch that one, then it will be so easy for you to understand this topic. Okay, so let's begin our the today's discussion is about nucleic acids. Basically, nucleic acids contains two molecules. Nucleic acids contains two molecules. Nucleic acid contains two molecules. The first molecule is RNA and the second molecule is DNA. So now let's discuss about RNA and DNA. So RNA stands for ribonucleic acid, and DNA stands for deoxyribonucleic acid. Okay, now just look at this. As in the previous video, I discussed about the RNA and the DNA molecule. So how we can get the RNA and DNA molecule? If we look at the structure of the RNA and the DNA molecule, we will find that in the RNA molecule, the RNA molecule, the sugar molecule in RNA structure is ribose, and this sugar molecule is pentose. Pentose means it's basically a 5-carbon ring structure. So same as in the DNA molecule, the sugar is also the same. It's ribose, and the sugar molecule is same as here. It's also a 5-carbon. So here it is also a 5-carbon ring structure. Okay, now let's draw the structure for the RNA molecule so that then you can easily understand how we can get DNA molecule from the RNA. Okay, so just now look at the structure. So this is an oxygen molecule, and here is okay, wait. Okay, just look at this. It's oxygen molecule. Here is carbon number 1. Here is carbon number 2. Here is carbon number 3. Here is carbon number 4. And here we go with carbon number 5. So if we the structure I draw is little irregular, just let me draw. It's an oxygen molecule, and here we go. Okay, now look at this. It's a 5-carbon ring structure. The carbon number 1, 2, 3, 4 and the 5. So on the carbon number 2, you will find that there's an oxygen molecule attaches to it. There's an oxygen molecule attaches to the carbon number 2. And in the previous video, I told you about that. On the carbon number 1, you will find that there is a nitrogenous basis always attaches on the carbon number 1, and the phosphate molecules can either attach on carbon number 5 or they can either attach on carbon number 3. So basically this structure is about the RNA molecule. Okay, now let's draw the structure. For DNA molecule. So here is the oxygen molecule. It's carbon number 1, it's carbon number 2, it's carbon number 3. Here we go with carbon number 4, and here we go with carbon number 5. So same as the nitrogenous base always attaches to nitrogenous bases attaches. Sorry, I just did a little mistake. Nitrogenous bases attaches on carbon number 1, and the phosphate molecule attaches on carbon number 5, or they can either attach on carbon number 3. The little thing which differentiate between RNA and DNA is that in RNA you will find that there is an oxygen molecule attaches on the carbon number 2, and in DNA molecule there is no oxygen molecule on the carbon number 2. Okay, so our basic today's discussion is just about the DNA molecule, because our genetic information is stored in DNA molecule, and then that genetic information we pass in our next generations. So in today's discussion we will not discuss about the RNA because that's a little simple and easy, so we will just focus on the DNA molecule. Our today's mainly discussion is about DNA molecule, is on DNA molecule. Okay, so now first I will explain you some basic characteristics of the DNA molecule, and then I will help you to understand DNA molecule from the structure way, okay, from its structure. So if we look at the DNA molecule, so DNA molecule is basically a double-stranded DNA molecule. It's a double-stranded DNA molecule. Okay, what does I mean from the double-stranded molecule? Okay, now just let me explain it to you. The double-stranded molecule is, just suppose like this. If this is a one strand, so just here is the second strand. If this is the first strand, then this is the second strand. Okay, so now just look at this. The double-stranded DNA molecule, or in the short form you can write it double-stranded DNA molecule. Okay, so basically in human beings, in not only just in human beings, in some other species, in some bacteria, or some in the species from the kingdom Animalia, the DNA molecule is mostly double-stranded DNA molecule. But there are some species of viruses, there are some viruses in which the DNA molecule is not double-stranded In some viruses, the DNA molecule is single-stranded like this. Is just like this, the single-stranded. Okay, just like this, the single-stranded. So what kind of viruses these are? In some viruses, the DNA molecule is single-stranded DNA molecule is single-stranded in some viruses. Okay, now what kind of viruses are they? The most common example of this is parvovirus B19. So this virus, parvovirus B19, contains a single-stranded DNA molecule. Okay, as we are just discussing about this virus, so here is a small pathology related to this virus. Basically what happens is that this parvovirus B19 is attack on our the bone marrow, where it goes with our bone marrow. This is the bone marrow. So this parvo B19 virus attacks on our bone marrow, and then it start start attacking on the bone marrow, and then we get a disease called as aplastic anemia. Okay, so here is a little pathology related to this virus. So this parvovirus B19 attaches on the bone marrow, and then the bone marrow, the bone marrow produces the stem cells for the blood cells and some other cells. So then we can get the aplastic anemia just because of this virus. Okay, so the DNA molecule is double-stranded, and in some species of the viruses, the DNA molecule is not double-stranded; it's single-stranded. And the other thing, the other characteristics for the DNA molecule is like that. The DNA molecule is a double helix structure. It's a double helix structure. It's a double helix structure. Okay, what does I mean from the double helix structure is like that. If you can look at this, if this is the one strand, then the second strand is like this. So the second strand is like this. So just look at this. This is the first strand. If suppose this is the first strand, this is the second strand. So they are, they just make coils. So they are in a double helix structure. The third thing about the DNA molecule is that it's a polynucleotide structure. It's a polynucleotide structure. It's polynucleotide structure. Okay, what does I mean from the polynucleotide structure? Poly means anything which is more than one, so we call it poly. So nucleotide structure. In the previous videos, I told you about the nucleotide structure. Okay, now here I will let you know what does I mean from the nucleotide structure. Okay, just look at this. Just now, look at this. We will first make a nucleotide structure. So here we go. This is our sugar molecule. This is our sugar molecule, and on the sugar molecule there is a nitrogenous base attaches to it, and at the same time there is a phosphate molecule attaches to it. Okay, so we call this a nucleotide. So we call it what? Nucleotide. This is our nucleotide structure. So here I discuss about the polynucleotide. Means if this is the one nucleotide structure, okay, suppose here we make another, this is the sugar molecule, and here is the nitrogenous base, here is the phosphate molecule, this is the second, okay, just suppose here we make another one, here we make another one, here we make another one. This is a sugar molecule, this is the nitrogenous base, and here the phosphate molecules. So just look at this, when there is when there are some bonds in between these nucleotide structures, like suppose this and this, so then we call it the polynucleotide structure. Just after a while I will make the proper structure for the DNA molecule, and then you can understand what does I mean from the polynucleotide structure. Okay, so who basically proposed this DNA molecule? Who proposed about the DNA structure? It is basically called as Watson and Crick. Double-stranded DNA molecule. So basically this is the Watson and Crick double-stranded DNA molecule. Okay, so now we will just focus about the structure of the DNA molecules, and then I will let you know some more basic points and some more important points about the DNA molecule is that whenever you find there are base pairs in your exams or in your books, and if the writer or the examiner don't tell you that the directions how you can read or how you can synthesize a DNA molecule, so basically you just have to examine, you just have to imagine that the DNA molecule we always read in 5 prime and to 3 prime end. Okay, this is an important line, this is an important characteristics of the DNA molecule, Sometimes in the books or in the exams, the examiner or the writer will not let you know that how you can read or how you can synthesize the DNA molecule. So you just have to suppose that if there is nothing, there is no information about the DNA molecule, so you have to imagine that you have to synthesize or you have to read that code or that base pairs in 5 prime end to 3 prime end. And if there is the writer told you, then it would be either in 3 prime end to 5 prime end. This is if the base pairs of the codes written in the book or in the exams and the writers are 3 prime end and 5 prime end, then you don't need to suppose that from the 5 end or 3 end, okay? But if they don't tell you how you can read or how you can synthesize the DNA molecule, so then you have to imagine and you have to suppose that you always read and synthesize the DNA molecule in 5 prime end to 3 prime end, okay? How you can make or get DNA molecule. First, I just told you that DNA is a polynucleotide structure, and okay, so here we go with our the first nucleotide structure is just suppose there is a sugar molecule. Suppose there is a sugar molecule and the sugar molecule is oxygen. Here we go with our carbon number 1, carbon number 2, carbon number 3, carbon number 4, and here we go with carbon number 5. Okay, this is our first nucleotide structure. But as I told you that the our nitrogenous bases can I always attach always our nitrogenous bases attach on carbon number 1 and our phosphate molecules attach on carbon number 5, or they can either attach on carbon number 3. So basically this is the first nucleotide structure. Okay, let's draw another nucleotide structure. If this is an oxygen molecule, here we go with carbon number 1, here we go with carbon number 2, carbon number 3, carbon number 4, and here we go with carbon number 5. Okay, so look at this, the nitrogenous base attach on carbon number 1 and the phosphate molecule attach on carbon number 3, or it can also attach on carbon number 5. It can also attach on carbon number 5. But now here I'm just I will explain it to you why I'm not writing the phosphate molecule here. I will just explain it to you, okay? So please just be patient for a while. Okay, now this is these are I just draw two nucleotides. So this is a one strand of the DNA molecule. Let's draw for the same nucleotides for the second strand of the DNA molecule. Let's draw it for it. This is an oxygen molecule. Here we go with our carbon number 1, carbon number 2, carbon number 3. Here we go with our carbon number 4, and wait. Here we go with our carbon number 4, or here we go with our carbon number 5. Okay, now the nitrogenous base attach on carbon number 1, or the phosphate molecule can attach on carbon number 3, or the phosphate molecule can attach on carbon number 5. Now let's draw the second nucleotide for the second strand of the DNA molecule. Here is a DNA molecule. Here is an oxygen. Here is an oxygen molecule. Go with carbon number 1. Here we go with our carbon number 2, carbon number 3. Oh wait. Now we have to make a bond in between them. We have to connect the two nucleotides. So let's draw from there. It's an oxygen. carbon number 1, carbon number 2, carbon number 3, carbon number 4. Here we go with our carbon number 5. Okay, so same, the nitrogenous base attach on the carbon number 1, and the phosphate can either attach on the carbon number 3 or it can either attach on carbon number 5. Okay, so I just wrote, the DNA is a double-stranded molecule. So for the DNA, the strands are double-stranded. So this is the second strand, and this is the first strand molecule. Okay, so now just look at this. Now just look at this. This is the first sugar molecule. This is first sugar molecule. This is second sugar molecule. This is, this is also the first and this is also the second. Now look at this. This is the first sugar molecule, and this is another sugar molecule. So the first sugar molecule of the first nucleotide attaches to the carbon number 5 of the second nucleotide of the sugar molecule. Look at this. So here we go with our, the carbon number 3, the phosphate molecule attaches with the carbon number 5 phosphate. So basically this kind of bond, so this kind of bond is called, we call it phosphodiester bond, or the linkage. You can either call it phosphodiester bond, or you can either call it phosphodiester linkage. Basically, this is the backbone of the DNA molecule. How? Just look at this. This is the first nucleotide, and this is the sugar molecule of the first nucleotide. And here we go with the second nucleotide. This is the, the sugar molecule of the second nucleotide. So, the carbon number 3, of the first sugar molecule, contains a phosphate molecule. And here we go, the carbon number 5 of the second sugar molecule have this, the phosphate molecule. So when the phosphate molecules, they attach with each other, and in this way the two sugar molecules can link together. And basically when this is just 2, so maybe it can either, the 3 and the 4 and the 5, so multiple, the nucleotides, they can just link with each other, just making the phosphodiester bonds. And here we go, just look at this. And here, this is the carbon number 5, this is the carbon number 3. So this is the, the first nucleotide, this is the second nucleotide. So here the carbon number 5 molecule, the carbon number 5 contains a phosphate molecule, and the carbon number 3 also contains a phosphate molecule. And when they link together, they also make a phosphodiester bond. They also make a phosphodiester bond, or you can either call it phosphodiester linkage. So this is basically, in this way, by making when the carbon number 5 phosphate molecule and the carbon number 3, so when they attach with each other, so the two nucleotides, they get attached with each other, and in this way you can make a long structure, you can make a long strand for the DNA molecule. Okay. So just now we discussed about this, and now I will let you know that here is the carbon number 5 of the first sugar molecule contains a phosphate molecule, and this is the free end. This is a free end because there is no other nucleotide molecule attaches to it, so it's a free end. So there is nothing attached to this phosphate molecule. So we call this one, the 5 carbon. So it is the 5 prime end. This is the 5 prime end. And just look at this. This is the carbon number 3. So here the phosphate molecule is also free. There is nothing attached to this. So this is the 3 prime end. This is 3 prime end. So this is our first strand molecule, and as I told you before that we always read or synthesize the DNA molecules from the 5 end to 3 prime end direction. If this is the first strand, so now just look at the second strand. Now just look at the second strand. Look at this, look at this. This is the carbon number 3, and this phosphate molecule is free. There is nothing, there is no other nucleotide is attached to this. So this, the carbon number 3, so this is the 3 prime end is free. And look at this is the carbon number 5. It's also free. There is no other sugar molecule attached to this. So this is, the 5 prime end. So as I told you that we always read or synthesize the DNA molecule from 5 prime to 3 prime end, from 5 prime end to 3 prime end, from 5 prime end to 3 prime end. Okay, so that's I guess not so difficult, so that's so simple. You can just look at the video maybe one more time. If you don't understand, then I hope you can just understand what I just told you. So I just told you about other things. Now let's discuss about the nitrogenous bases. Now let's just discuss about the nitrogenous bases. There is a rule called as Chargaff's rule. Chargaff's rule. So basically what this rule told us, that the nitrogenous bases is basically composed of purines and the pyrimidines, and the purines further classified into adenine and guanine, and the pyrimidines contains the cytosine, uracil and the thiamine. So now we are discussing about the DNA molecule. So in this we will not talk about the uracil, we will not count the uracil, but we will just count cytosine and the thiamine. If we talk about RNA molecule, so then we will consider not thiamine, we will consider the uracil molecule. Okay, so in this Chargaff rule, basically what this rule told us is that adenine always goes with thiamine and cytosine goes with guanine. Adenine goes with thiamine and cytosine goes with guanine. What does it mean? Basically, adenine and thiamine, the ratio or the amount of these bases should be equal, and cytosine and guanine, the ratio or the amount of these two bases should be equal in the DNA molecule. Look at this. In between the adenine and thiamine, the hydrogenous, these are the hydrogen bonds. In between adenine and thiamine, the hydrogen bonds are 2, and in cytosine and guanine, the hydrogen bonds are 3. Okay, so look at this. If just suppose that if this one is, okay, just suppose if this is adenine, then here we go with thiamine. Okay, suppose it's cytosine, then here we go with guanine, and the bond in between them is double, and here it's 3 hydrogen bonds in between cytosine and guanine, and 2 hydrogen bonds between adenine and thiamine. So our the basic the little information about the DNA molecule is that the DNA molecule contains 2 strands. The DNA molecule contains 2 strands. So these are the 2 strands. This is the first strand, this is the second strand. Okay, so we always read from 5 prime end to 3 prime end. Okay, 3 prime end. So same, if so the two strands are complementary, the two strands are complementary and anti-parallel, and anti-parallel. Okay, so if this is the 5 prime end, so here we go with our, if this is a 5, so here we go with our 5 prime end, and it's here 3 prime end. Okay, look at this, 5 to 3 direction and then 5 to 3 direction. The last thing about the DNA molecule is that how you can denature the DNA molecule. What are the things which can denature the DNA molecule? Normally our DNA molecule is like this in a double helix form. Normally, it's like this. So this is our the DNA molecule, the double-stranded DNA molecule, which is in a double helix form. They are making coils. So what are the things? What are the things which can denature the DNA molecule? So the first thing is heat. If you start heating the DNA molecule, it can denature the DNA molecule. Or the other thing is alkaline pH, or the other thing, it can be the chemicals. Okay, so if this is a double-stranded DNA molecule, now these are the things which can denature, the DNA molecule, denature DNA. Okay, so now look at this, the two strands, now they are connected with each other, from the phosphodiester bonds, and in between the base pairs there are hydrogen bonds. So upon heating on an alkaline pH or with some chemicals, the DNA molecule, it's like this. This is the first strand and this is the second strand molecule. So now if you again want to renature the same DNA molecule, so what you can do, you heat it, or you try to give some heat on to the DNA molecule. So upon cooling, you can again get the same DNA molecule. Okay, you can renature. DNA molecule. So this is our today's discussion. I hope you guys will like it, and if you guys like it, just please give a big thumb up. So inshallah I will catch you in the next video. So have a good day. Bye bye.